Let me start by clarifying what's interesting about sperm chemotaxis in turbulent flows. First of all, chemotaxis describes the navigation of biological cells guided by chemical gradients. For instance, sperm swim on helical paths in the search for a target egg. The egg releases a chemical called chemoattractant, shown in blue here, and via helical chemotaxis, the sperm is able to steer up this chemical gradient and successfully navigate towards the egg. Sperm chemotaxis has been intensively studied for the external fertilization of marine invertebrates, for instance, sea urchin and red abalone. But it has also recently caught attention in the context of drug delivery. The mechanism of sperm chemotaxis is well established in the literature, both on a theoretical and physiological level. In fact, there exists a model to simulate sperm behavior numerically. However, this work focused predominantly on idealized conditions of perfect chemical gradients in still water. Yet natural environments, such as the ocean, are characterized by turbulent flows, which distort the concentration fields. Such distortions have been studied in the context of scalar turbulence and turbulent mixing. For instance, consider a cloud of sperm and eggs shown in red and blue here, which mix in the turbulence created behind this black obstacle here. Note, however, that this turbulent mixing actually underpredicts sperm-egg encounters, and a major reason is that the navigation of sperm cells is neglected here so far. So there's a gap of knowledge in between the small-scale still water regime and the large-scale turbulent mixing regime without chemotaxis. Moreover, experiments that have been conducted in this regime revealed that there's an optimal turbulence strength, which is not yet understood. So, for instance, here on the y-axis, you have some measure of the sperm-egg encounters, and on the y-axis, you have some measure of the turbulence strength, where 0 corresponds to still water, and you can see we observe the maximal sperm-egg encounters at a non-vanishing turbulence strength. And while it is not understood for sperm, note that similar optima has been observed actually for bacterial foraging by Taylor and Stocker. Now, I'm going to show you how we recover such an optimum in numerical simulations, as shown by this blue curve here, by applying the chemotaxis model to turbulent flows. I'm going to further characterize how sperm surf along the filamentous concentration field, which is typically generated by shear flow present. Finally, I'm going to use the this insight to semi-analytically estimate the position and height of this optimum, as shown by this red curve here, from which we conclude that the origin of this optimum is actually the competition between faster establishment of concentration gradients versus the distortion of these gradients. Finally, we compare our simulations and theory to known experimental results. We first have to decide how to model sperm chemotaxis in turbulent flow. So this here is a sketch of a typically turbulent flow. We have some initial eddies or vortices, which decay over time to smaller and smaller eddies until these are so small that they dissipate and vanish. If we put an egg into such an eddy, the corresponding concentration field may look like this. So in the center here, you have the egg releasing the chemoattractant. And as you can see in the turbulent flow, the chemoattractant forms these patchy and filamentous concentration fields. Now, the scales of these structures are mainly determined by the Kolmogorov scales. For instance, the thickness of the patches or size of gradients is given by the Bechdel length, and the size of the smallest eddies by the Kolmogorov length. Now, if we compare these spatial scales, it turns out that sperm chemotaxis takes place on much smaller scales than the smallest eddies. And it is known that on such a small scale, turbulence is actually perceived as a simple shear flow. In conclusion, for our purpose, this means that it is sufficient to consider a simple shear flow, and we do not need to simulate full-scale turbulence. So we already know the first component of our simulation framework, the flow field. The second component is the model for sperm chemotaxis from the literature. That is a helical swimming path, which is modulated by a chemotactic signalling system. The last component is the interaction with the flow, which we implemented via convection and co-rotation of the sperm, where we assume sperms to be prolate spheroids. If we combine these three components, we get, for instance, such a turbulent concentration field. And in this concentration field, we get this kind of sperm trajectories, where we see that sperm hit the filament and then kind of surf along it. Some going towards the egg, some going away from the egg, some getting stuck in these patches. And the important part is that this filament surfing is also observed in the much simpler case of a shear flow. So we're going to focus on this case in the following. So let's compute the fertilization from the simulation. We assume some volume around the egg with periodic boundary conditions and distribute initially the sperms uniformly in this volume. And it turns out that we actually recover an optimum of the sperm-egg encounters in this simulation. In fact, the leftmost point corresponds to no flow, and as you can see, the optimum is about 8 times higher than just for normal diffusion. Furthermore, we get very similar results with and without co-rotation, meaning that we will neglect co-rotation in the following. Now the question is, how do we understand the origin of this optimum? The easiest explanation would be to say that this basically scales with the volume that the chemoattractant covers. However, for the given parameters, this chemotactic volume actually increases for all of these shear rates. So we actually have to examine the dynamics within the filaments to understand the optimum. For that, we characterize the far field of the filaments, and it turns out that the filaments can be described by this function with an Gaussian decay of the concentration away from the center of the filament and an exponential decay along the filaments. And for an idealized filament, this may look like this. So here again, we have the egg in black here, and now sample the concentration field on these cross sections. You have the Gaussian decay going away from the center of the filament, and then this exponential decay along the filament. And now if we inspect a sperm path in this concentration field, like this one, we see the helical path. Now the sperm senses the gradient, navigates towards the center of the filament, misses it, and then starts to oscillate around the center, in this case actually missing the egg on the first try, back here realizing its mistake, turning around, and then hitting the egg. How can we characterize this dynamics? I will skip the details of the derivation. We basically just apply linear response theory to the concentration field of the filament and obtain an ODE, which represents in first order just a damped oscillator around the center of the filament. Now we can compare this prediction, for instance, to the distance from the center of the filament over time for the sperm trajectory we have looked at. And in fact, we find a good agreement both between the decay of the amplitude as well as the frequency. And in fact, we find this for the majority of paths independent of their entry angle into the filament. The only condition, though, is that the filament is thick enough For instance, if we go further towards the end of the filament, we may get trajectories like this, which get close to the filament, feel it, but before they can make the first turn inside the filament, they already left the filament again. So these parts of the filament, in the sense, are not effective for navigation. If we not just look at the sperms that reach the egg in the simulation, but rather the ones that stay inside the filament, we actually find a nice match between simulation and the predicted effective volume according to our theory. And we find so also for the real filament. However, still, while we observe an optimum here, it's not at the same position and height of the optimum of sperms reaching the egg. And that is because we additionally have to consider that sperm needs some time to swim towards the egg after they're inside the filament. With this slide, I just want to hint at the details of the theory. You basically formulate an inner and an outer search problem. And the only thing I want to point out here is that this still leaves a parameter which we have to fit, that is the flux of sperm into the filament. However, we find this fitted value very close to an estimation you can derive for a ballistic swimmer. In this sense, the heuristical origin of the optimum is that on the one hand, higher shear flow spread the chemoattractant faster in form of filaments, which facilitates sperm navigation to the egg, but on the other hand, make these filaments longer and thinner, which inhibits this navigation. Now, having understood the optimum for numerical simulations, we could compare how well our simulations reflect actual experiments. So here is some data obtained by Zimmer et al. For Red Abalone, where they measured the fractions of eggs that are fertilized at certain shear. And they did it both with chemotaxis and without chemotaxis. And in order to compare this to our theory, we have to translate the fraction of sperm reaching the egg, which our theory predicts, to the fraction of eggs fertilized, for which you use a simple expression that basically just introduces an additional probability that a sperm fertilizes an egg at an encounter. And if we translate the experimental setup to our theory, we get a reasonable correspondence between our simulations and theory and the experimental results. Of course, there are deviations, as you have to remember that our model only presents a minimal approach. And I further want to hint at another experiment for strong turbulence, where there's additional data by which the fertilization probability can be calibrated independently. And also here we find a reasonable correspondence with both our simulations and theory. This brings me to my conclusions. We recovered an optimal turbulence strength in numerical simulations, described the sperm dynamics in terms of filament surfing, and explain and predict based on this dynamics the optimal turbulence strength. We find reasonable agreement between our simulations and experiment. Of course, our simulations represent only a minimal model, and in the future we have to take into account additional effects which occur in experiments like chemokinesis, turbulent mixing, as well as we can extend our model to more generic turbulent flows like unsteady shear flow or Burger vortices. And it would also be a good idea to apply or extend the model to bacterial foraging. So with that, I thank you for your attention.